In this video, we are going to talk about cellular respiration, and the very first step of cellular respiration is called glycolysis. Now, this is an anaerobic process, which means that it happens and oxygen does not need to be present for this. Because of this, it's very simple, and all organisms are capable of doing glycolysis, even if they don't continue on and do the more complicated steps of cellular respiration. And glycolysis occurs within the cytoplasm of the cell, so it does not require any specialized organelles or anything. So again, prokaryotes, eukaryotes, all of them can do glycolysis. So let's look at the word glycolysis. So this glyco part here refers to sugar or your carbohydrates, and then lysis refers to splitting. So during this phase, what the cell is doing is splitting up the sugars. And I'll show you how that occurs. So glycolysis is broken down into two steps. There's your energy investment stage, in which ATP is utilized in the reaction, and then there is your energy return when ATP is given back. So the first thing we need to do is represent glucose here. And remember, the chemical formula for glucose is C6H12O6. So we have 6 molecules of carbon. And in the very first step, this is where we're going to use our ATP. Now I'm going to draw a long line like this, because glucose, the 6 carbon molecule, is broken down into 2 3 carbon molecules. Okay. So before I show you that, we need to show that our ATP is going to be used, and of course that is kicked out, so you get ADP plus P, and those are going to be recycled and used later. And you need ATP for both of these. Okay, and that is called our energy investment. All right, and during the energy investment stage, our glucose, which is a 6 carbon molecule, is broken down into 2 3 carbon molecules called PGAL. And I'll show you the really simple version of the chemical formula for this. Essentially, it is a phosphate and 3 carbons. Okay. Now, PGAL is going to be converted into BPGAL. And both molecules of PGAL are going to do this. And during this process, you end up with molecules of NADH being formed. So this molecule requires NAD plus, oops, that kind of looks like a P, NAD plus, and that is going to come out as NADH. And technically you also have a plus H plus here, and that's going to be used later as well. But the whole key is that you end up with NAD, oops, I lost my whiteboard there, NAD plus, and we're going to get our NADH. All right, so BPGAL is 2 phosphates with 3 carbons here. Okay. Now in the next step, BPGAL is going to be converted to PGA, and in this stage you end up actually with your ATP being produced. So remember, to get ATP you take ADP plus a phosphate. This is like really hard to draw, all of this on one little screen. And now you get your ATP. Same thing happens down here, ADP plus P, and that is going to be kicked out as ATP. Okay. And PGA is 3 carbons with a phosphate as well. And for the very last step, PGA is going to be converted into pyruvate. And during that step, again, ATP is produced, so you have your ADP plus P, and that produces ATP. So you have ADP plus P being kicked out as another ATP. All right, so that is what we're looking at for glycolysis. So all of this stuff here then is going to be our energy return. All right. So when we're looking at our yields of glycolysis, it's just going to be very important to understand what the yields actually are. You have your 2 molecules of NADH. Notice that 4 molecules of ATP are created. However, 2 molecules of ATP were used in the beginning, so your net yield of ATP is actually just 2 molecules of ATP. Okay, so even though 4 ATP are created, you're only making technically 2 new ATP. And then finally you have 2 molecules of pyruvate. Now if oxygen is present, those 2 molecules of pyruvate are going to move on to the prep step, which will occur within the mitochondria. However, if oxygen is not present, then the 2 pyruvate can move on to the lactic acid cycle or alcohol fermentation, and those are processes that can still generate molecules of ATP, but you're not getting as much ATP, you're not getting your NADH, and that does not require oxygen. So in the next video, I will talk about the prep step, and then we will move on into the Krebs cycle. And then in the last video of this series, we will talk about the electron transport chain.